Hi there, my name is Andrew Brown, and welcome to Cell Cycle and Mitosis. This video will take you all the way through the cell cycle, starting with interphase and paying special attention to the four phases of division. As you watch, try to keep an eye out for the similarities and differences, both within interphase and during mitosis. After watching this video, you should be able to answer questions like this one, which asks you to compare and contrast the 5 stages of the cell cycle. We'll start by going over some general aspects of the cell cycle. Dividing cells fall into 2 main groups, diploid autosomal cells and haploid germ cells. The N number for each refers to how many genome copies there are in the cell's nucleus. Diploid have 2 and haploid have 1. While division is often the most emphasized in cell cycle biology, remember it's only one of 4 stages. The other 3, G1, S, and G2, are known collectively as interphase. Here, cells grow and the genome is duplicated so that mitosis and meiosis can occur, making interphase the longest part of the cell cycle. Cells that do not divide, such as muscles and nerves, never actually enter the S phase. They get trapped in a special phase called G0, which refers to continued growth. Now let's take a look at all 4 stages of the cell cycle in greater detail. G1, known as the presynthetic gap, is the phase in which organelles develop, the cells grow, and the first checkpoint is reached. More on those in just a bit. The next phase, S, which stands for synthesis, is defined by replication of the cell's DNA. After this, the chromosomes appear in their familiar X forms, which are really just 2 identical chromatids. G2, the post-synthetic gap, involves a couple more checkpoints. One to make sure there is enough cytoplasm and organelles for division, and another to suss out any errors created during genome replication. Then, finally, after all that, it's time for mitosis. Technically, mitosis refers just to the division of chromosomes in the cell's nucleus, so cytokinesis, the division of the cytoplasm, is also needed to complete the phase. Now that we've covered the basics of the cell cycle, let's talk more about those checkpoints that were mentioned earlier. The checkpoint between the G1 and S phases allows the cell to check the DNA for damage or mistakes. If any problems are found, the cell will wait for the DNA to be repaired before moving on to S phase. The next checkpoint, between G2 and mitosis, is to make sure the cells are big enough and that their organelles have developed sufficiently for division to occur. Some of the proteins involved in these checkpoints are p53, various cyclins and their dependent kinases, and transcription factors, which interact directly with the DNA during replication. Because the failure of those checkpoints often results in cancer, we will now say a little bit more about what is occurring in cancerous cells, and how the major players are involved. Basically, cancer is unrestricted cell division, stemming from the disruption of cell cycle controls. When the checkpoints are bypassed, cells with damaged genomes make it through to mitosis and are spread. A mutated p53 gene is a common finding in cancer cells. When p53 is inactive, damaged DNA does not get flagged at the appropriate checkpoint. Mutations accumulate as the cancerous cells keep dividing, and eventually a tumor forms if division is rapid enough. Metastasis occurs if cancer spreads to other tissues in the body. Okay, that takes us through the controls on the cell cycle and lack thereof. Now we'll explore in more detail the final phase of the cell cycle, mitosis. The object of mitosis is to create 2 identical daughter cells from a single parent cell. To be clear, this occurs in somatic cells only, not in cells of the germline. Mitosis also has its own distinct phases. The first is known as prophase, then metaphase, followed by anaphase, and finally telophase. We'll start by taking a closer look at prophase. At the end of interphase, the cell looks like this. The nuclear membrane is full of loosely packed chromatin, and the centrosome, with its centrioles inside, still resides in the center of the cell. As prophase arrives, some marked changes begin to occur. The chromatin condenses into thicker fibers wound around histones, centrioles migrate to the cell's poles, at which point microtubular spindle fibers begin to spring forth from the centrioles. The nuclear membrane dissolves, and the growing spindle apparatus makes contact with the condensed chromosomes. The points at which the spindles reach the chromosomes are henceforth known as kinetochores. The next phase of mitosis, metaphase, is best known by the image of chromosomes lined up at the middle of the cell. By now, the centriole pairs have reached opposite poles within the cell. Interaction between the spindle fibers and kinetochores positions chromosomes in a line on the metaphase plate. At this point, chromosomes are in the exact center of the cell, and the cell is ready to undergo anaphase. During anaphase, centromeres now split, releasing the two chromatids with their spindle fibers still attached. Each chromatid still possesses a centromere. When the chromatids break apart, they are still under tension from the spindle fibers and begin to migrate toward opposite poles of the cell. The baby steps of cell division are now underway. In the final phase of mitosis, telophase, the two daughter cells will now begin to form. It is basically the reverse of prophase. As the chromatids reach the cell's poles, the spindle apparatus disappears, the nuclear membrane reforms, and the chromosomes uncoil into less dense chromatin. At this point, there are essentially two nuclei in the cell, and it's time for cytokinesis. Cytokinesis is the division of the cytoplasm. It begins with a slight invagination at the edge of the cell on the metaphase plate and ends with two identical daughter cells. And that actually does it for the material this video will cover. Now let's apply what you've learned on a couple practice questions. This first one asks you to predict the phase of mitosis that is affected by a disease that alters how chromosomes line up at the equatorial plate. The answer should be D. Remember, this question is asking about the mitotic phase that is directly affected by the disease. While several phases might be indirectly affected by alterations at the equatorial plate, only metaphase will be directly affected. This next question is asking you for the statement that correctly describes the stages of the cell cycle. The answer should be B. Interphase is the longest phase, not the shortest. The restriction point in G1 ensures that the cell is ready for S phase, not mitosis, and the genetic material in a cell is changed from haploid to diploid during S phase of meiosis, not mitosis. Alright, if you got that question correct, congratulations. If not, please feel free to go back and watch this video again as many times as you need. Before we finish, let's take a moment to review what was covered in this video. The cell cycle consists of G1, S, G2, and M phases, plus G0, the special offshoot of G1 reserved for non-dividing cells. We saw two checkpoints in the cell cycle, one between G1 and S to check the DNA for damage, and another between G2 and M to make sure the cell is ready for division. Cancer often results from broken checkpoints, allowing division to occur unchecked and uncontrolled. The mitotic phase consists of its own four phases, prophase, metaphase, anaphase, and telophase, and is followed by cytokinesis, during which the separation of the binucleate parent cell actually splits into two daughter cells. Thanks very much for watching, and good luck in your continued MCAT study.